Alright, picking back up with lysosomes. So lysosomes are individual membrane sacs, and inside of them it has these hydrolytic enzymes. Remember, most of our biological macromolecules are formed with dehydration synthesis. So we remove a water molecule to put various monomers together to form a polymer, and then hydrolytic enzymes would work to break those bonds. Now, if you recall that this would involve adding a water molecule, this actually works better when you are in an acidic environment. So the inside of this lysosome is both acidic and has all these hydrolytic enzymes that break down macromolecules. You could see how this would be a dangerous compartment for a cell. If it were to leak open, it would release that and it would start eating itself. In the same way, if we put a hole in our stomach, we would start to digest ourselves from the inside out. So these little vesicles act as the stomachs of a cell. Now, all those hydrolytic enzymes and that membrane material are essentially made in the rough ER, transferred through the Golgi apparatus, and along the way, these digestive enzymes are processed until they are eventually activated. You could imagine that if you had an enzyme that would break down macromolecules, you would need a safety on that. So when the protein is first made, it is not functional. Usually it is a part of that protein needs to be cleaved off or removed before it becomes active. And this process of transferring it through the Golgi apparatus and in through vesicles to other things, ultimately to the lysosome, is part of the process is part of the process of activating those molecules. Now, sometimes when a cell encounters material that it wants to consume, it is in too large of chunks for us to break down and absorb through our plasma membrane or to import through channels. So typically, channels that go through our plasma membrane are only bringing in simple sugars, amino acids, very simple supplies. So if we have anything bigger, what we need to do is essentially incorporate that material through a process that we call phagocytosis. So we get an invagination of the plasma membrane that draws in the material. It pinches off into what we call a food vacuole. We can then take our lysosomes and fuse them with these food vacuoles, breaking down the material inside. Then we can absorb those individual monomers that can be pulled in through specific transporters. Now, in addition to bringing in food from the outside, we can actually utilize a very similar process to recycle some of the cell's own material. So if we have larger organelles that have ceased becoming functional, we can actually envelop those in its own separate food vacuole and then digest that material. And that is something that we call autophagy. So here, for example, we have an old mitochondrium and we have an old peroxisome, which we haven't talked about yet, but we'll get there. And both of these have ceased to be functional. So we envelop it in its own vesicle that we can then fuse with the lysosome and break that material down, recycle all the pieces, and build new ones. So, concept 6.4, the endomembrane system regulates protein traffic and performs metabolic functions in the cell. So we have all these separate compartments that specialize in different activities. We have the smooth ER, and the rough ER, which is continuous with the nuclear membrane. Smooth ER, we can produce various lipids, and we can carry out breakdown of toxic materials. And then those materials can transfer on out. Here we have ribosomes attaching to the rough ER. It'll be attached to a messenger RNA, and it will produce a protein and inject it straight into the lumen of the ER. That protein can then start getting modified. Maybe you put on sugars, which act as address labels, and you can start putting them into vesicles and transporting them through the Golgi apparatus. You come in the cis side, which is closest to the nucleus. They can come out either the sides or through the trans side, and the ones coming out the trans side typically go to the plasma membrane. All along the Golgi apparatus, we can even make further modifications, slowly activating some proteins that might be digestive in nature that we can ultimately shunt into lysosomes, protecting the rest of the cell. So this transport system is something that we call protein traffic, right? So if you produce a protein in the cytoplasm, it will stay in the cytoplasm. If you want to put a protein somewhere specific, like let's say we want to put it into this compartment of the Golgi apparatus, it needs to come through this pathway. If we want to export it, it needs to come through this pathway. If we need to put it inside of any container, it essentially comes through this pathway. In having all these little compartments, we can essentially break up and concentrate various enzymes that do particular metabolic functions into different spots. So we could be breaking down drugs, we might be activating proteins over here, we might be adding address labels over here. Each spot has its own function. And that is the endoplasmic membrane system.